This presentation will be about management of spotted-winged Drosophila in blackberries, and a second one will be about broad mites. And the first one is results of Lizzie Herrera's research on spotted wing in Arkansas. The second one will be by Jessica LaFleur's. Spotted wing distribution started in California in 2008. 2009 it moved, they were found in Florida. 2010 you can see it moved up the coast into the Carolinas and Mississippi. By 2012 you can see it hit Arkansas, Texas, and many of the central states. And now, as of this year, we basically find it in any state that has fruit. Spotted wing Drosophila fact sheet that we distribute to growers includes information about how to identify a male, has a black spot on its wingtip. The front leg, here you can see these, a pair of what we call combs, two black combs on the front leg. Here's what the fly looks like as an adult male. And the female is very similar to the male, but it doesn't have spots on its wings. And it also has right here, this is the ovipositor or egg-laying apparatus that she uses to insert eggs into fruit. And here you can see a close-up of that sclerotized or hardened ovipositor. She actually saws into the skin of the fruit, and then she's able to lay an egg underneath the skin. On the left column you see spotted wing larvae in blueberry, in raspberry, these little white larvae, and in strawberry. Biology of the spotted wing: it spends about 1 to 3 days as an egg. Here you can see the white egg with these little thread-like spiracles. These are the breathing tubes, and they usually stick outside of the fruit. And here down below you can see the breathing tubes on the outside of a blueberry. The rest of the egg is inside in the flesh. Here's a larva that's come to the outside of the berry, and it takes 5 to 7 days to develop as a larva. Then it becomes a pupa, looking like this. It has a couple breathing tubes on the end, and that lasts for about 3 to 15 days. In terms of biology, they lay eggs. Here you can see on a blackberry drupe, the egg is inserted in the drupe itself, and it has these 2 breathing tubes outside. Life cycle takes 9 to 22 days. There's probably 5 or more generations per season in Arkansas, because we tend to start seeing them sometime in May, and we continue seeing them until frost. Here you can see some male flies on a strawberry. There's a spotted wing, probably the female with no spot. And they're active for 3 to 9 weeks before they die. So that's a long time to lay eggs. Female lays 7 to 16 eggs a day. So you can see probably a couple hundred eggs that are laid by any female during their lifespan. Signs of infestation: you'll get discolored spots, maybe on grapes. We don't have too much problem on grape, but this just shows in Oregon they have some problem on certain types of grapes. On blackberry, you can see we start to get limp hanging fruit, and the fruit cluster kind of dries out if you don't remove them, and sometimes you can actually see larvae on the outside. They tend to get watery inside. How we trap them is using a deli cup, 32-ounce deli cup, with the upper rim has about 20 of these holes that are about 3/16 of an inch in diameter. That's about just a little bit bigger than the fly itself, so you can see the flies are not very big. Inside, we take the cover off, and you can see that we cut a hole in the middle of the cover so we can put this wire to hold this lure inside the cup. And then we close it. We add some apple cider vinegar as a drowning solution for the flies that come in through the hole. And Century is the company that sells the spotted wing Drosophila bag lure, and it's available at Great Lakes IPM, as are the traps. But you can make these traps fairly easy. It's basically a deli cup that you get like potato salad in. Placement of the traps and collecting spotted wing Drosophila flies from the traps. Here you can see in all the flowers in blackberries, and here you can see the trap is set at the same height as the flowers and fruit. And we usually put the trap out when we see things starting to maybe have some green fruit. We tend to monitor the whole season for our research, but generally you can put these traps out about 3 weeks before you expect fruit to ripen. On strawberries, we put it right on the raised bed at the same height as the fruit. And here you can see the trap itself. We have usually a black line and a red line because they seem to be attracted to both those colors. Then you notice there's no holes on 1/3 of the trap. This is so we can pour the flies in the vinegar out into this little kitchen sieve, and we try to pour the vinegar into a catch container so we don't, and then we'll go throw it outside the planting. We try not to dump vinegar inside the planting because it'll attract flies just to that vinegar. But we remove the flies and put them in a little vial, and then we count them to see if there's any of the spotted wing. We can also monitor for eggs. This usually requires a microscope to see these little holes in the fruit and in the spiracles, so you can actually see if any of your fruit are infested. What we do is take 30 harvested fruit and check for the larvae or the eggs. You can actually use a 20x hand lens. And if you want to see if there's any larvae in the fruit, what we do is soak the fruit for about 30 minutes in a quart of water that is mixed up with a 1/4 cup of salt or 3/4 cup sugar, and this makes the larvae float to the surface and the fruit stay below. So you can actually see the larvae coming to the surface. We use this screen just to push the fruit down because some of that floats. Conventional control, if you're going to be putting on insecticide, especially in your small patches, we use some kind of backpack air blast sprayer to get good coverage. We have our boots and maybe a respirator if you need it, and a tie-deck suit. And generally it requires weekly applications of insecticide from first ripening fruit to last harvest. That means you need to alternate your treatments by mode of action to prevent resistance. And it's difficult to control high populations of spotted wing, so you want to start early. This slide was generated or assimilated by Isaac Rufus out of Michigan State. He basically asked across the country what compounds were working against spotted wing. And what he is looking for is, he categorized each of the compounds as either no activity, weak activity, fair activity, good activity, or excellent on the y-axis there from 0 to 4. And anything that was ranked as good or excellent above this dotted line, these are the compounds that we generally recommend. So you can see Asana, by Fincher, Brigade, Danitol, a lot of pyrethroids. Delegate, which is spinosad. Diazinon, which is an old OP. Exeril, Hero, Imidin, Lanite. Lanite's very toxic, so be careful with that one. Malathion, Mustang Max. You can see Radiant and also Warrior. In our fact sheet, we have recommended insecticides as well as the IRAC code or the mode of action for each of the insecticides. PHI is pre-harvest interval. That's the number of days after you've sprayed that you have to wait before you can pick the fruit. And then also the last column shows how effective that application is against spotted wing. So if you go by IRAC number, you can see the first ones, carbaryl and methomyl, are going to be in category 1. And a similar category of OPs are Imidin and Malathion. They're the same mode of action. Then you get in, all the 3s are the pyrethroids. The 5s are the spinosad compounds. And then the last one, number 28, is Exeril, which is a fairly new compound. Then I have information on blueberry, caneberry, and strawberry. Most of our damage in Arkansas is by caneberry. And these are the PHIs. And generally growers pick compounds like bifenthrin that has a 3-day PHI, or maybe malathion. They avoid things like carbaryl or 7 because it has a 7-day PHI. That means you have to wait 7 days to pick. Mustang Max has been a favorite. Delegate has been a favorite. Spinosad is a little bit expensive, but it could be used because it has a fairly good PHI. And you can see all these are ranked as excellent or good. Pyrethrin, which is a OMRI or organic compound, is only fair. So the only other OMRI compound we have is spinosad or Entrust. And it's good, but we hate to use it over and over and over again, but that's all we have for organic. Organic control, so Organic Materials Review Institute, that's what OMRI stands for. It reviews products that will be used or can be used in organic production. Like I said, the insecticides we have available for organic control is Entrust. So basically, we work on cultural practices, and for organic, we recommend that they early harvest, or at least harvest every day, and also sanitation, pick up all the fruit off the ground, and also take off cull fruit that are on the planting to minimize reproduction of and buildup of numbers of flies. Also, we're in Arkansas and also Michigan and Minnesota, we're testing exclusion. This is fairly expensive. You can see the ProTech Net Insect Exclusion screen, which is on these multi-bay tunnels, this one high tunnel on the sides and on the ends, and these little experimental screen tunnels. It costs a fair amount to get a 14-foot by 328-foot piece of screen. But it does work. So exclusion netting, it does lower percentage SWD infested berries, and we've proved that in several states. But it does, it's also successful in excluding some of the beneficial insects and also maybe the pollinators. To show you how effective it is, here we have our seasonal counts, biweekly counts of number of SWD adults captured in traps. And this is outside the wooded area, which is adjacent to the planting. Then outside our field row of blackberries, the traps are set. And we didn't have any get in the actual tunnel itself, so they're not represented in this slide. But you can see the woodlands, the green line indicated a peak in late June, and then much higher counts in July and August. They just keep building up in July and August and September. And the black line is outside the field row of blackberries. We had some harvest in July. Then we had most of our harvest was in mid and late July. So that's when we have a peak at that point. And 0 spotted wing flies in the screened high tunnel or low tunnel. Okay, this slide is similar to the last one. It shows percent of SWD infested blackberries. This is our 30 berry sample that we take every week. And you can see the first place to show any kind of damage is in early June, and that's in the field row by the screen and plastic tunnels. We had some damage up to about 30%. Then it kind of dropped down. Our harvest, we kind of went there wasn't any fruit out there for a while. Then we started getting the field row by the high tunnel. This was a little bit over to the west of the other site. And you can see in both cases in July we were catching outside on the blackberries flies. And then it really started increasing in August again in both the field sites. Then in late August we had a windstorm that opened up some of our screen tunnels, and one of the tunnels, of the 3 tunnels we had, the door got held open and it got some spotted wing in there, and we started getting infestations of spotted wing. But you notice from the previous slide we didn't have any capture of flies in the traps inside those tunnels, but we did. They were in there long enough to lay some eggs, probably in the morning, and then they left the tunnel during the day because it got too hot in there. We've noticed there's alternative spotted wing hosts nearby our commercial plantings. The main ones, some of the non-crop wild hosts include honeysuckle, wild blackberries, and pokeweed. And right now, pokeweed generally starts maturing in mid to late July. And these are important for some of our late summer spotted wing Drosophila buildup. Just to show you how the perimeter landscape affects your cropping area, this is Dripping Springs, which is an organic farm. And this shows seasonal total trap catches for 2016, and they had blueberries here in this area here where that gray bar is, and that's our total summer catch of spotted wing flies in traps in and around the blueberries. But you can see the orange bar to the north, the green bar to the south perimeter, and the pink bar to the east perimeter all had many more flies during the season than inside the commercial area. So it just shows the importance of maybe cooler environments in the edge of the planting, more shade, and also maybe some alternative food sources. So some of our best spotted wing management practices: thin or open up canopy to improve spray coverage. That's the biggest one. You don't want a really thick canopy, otherwise you can't get control of the flies that are hiding in the cooler shaded interior. Monitor traps for flies starting 3 weeks before ripening. Check traps weekly to see if you're catching anything. If found and fruit are starting to ripen, apply row sprays every 5 to 7 days with the recommended insecticides that I've already mentioned that we know are effective. And reapply after a 1-inch rain, because it probably washes off the insecticide. And then try to rotate to different modes of action. That's that IRAC number that I showed. And this is to delay resistance development. Evaluate spray effectiveness. In other words, after you've sprayed, go in a few days later and see if you have any eggs or larvae in the fruit. If you do, you might check to see if you need to improve your spray coverage, maybe go slower when you're applying, or change to a different compound. Exclude SWD using screen or row covers. That can be a possibility, maybe if you're a smaller acreage planter or grower. Pick daily and refrigerate, because once you put these fruit in the refrigerator, it'll kill the eggs in about 2 days. So it keeps the eggs from developing and emerging as thlarvae. Sanitation is also important. Pick leftover and damaged fruit and solarize those in a black bag, and then throw them in the garbage. And this helps reduce the spotted wing food source and also reproduction. And just stay informed, because like I said, our first time we ever saw this pest was 2008, and probably every state has someone working on it. So we're coming up with new management approaches every year. Thank you. I also wanted to thank the USDA for providing funds for our studies, and the Organic Research and Education Extension Initiative. We have a project that works on the organic options. We had several grad students and workers, and also several grower collaborators. The next part I'll talk about is management of broad mite in blackberries. This was work done mostly by Jessica LaFors, my master's student, and myself. Broad mite management requires that you ID both the damage and the mite itself. Here you can see the damage to the right. You can see the leaves compared to those leaves in the back that are kind of thin looking and nice and dark green. In the foreground you see these more thick leaves. They're starting to cup upward, and sometimes they'll cup downward, but they're also not as green. They're more yellow. They're pretty obvious compared to the healthy leaves. Here's the mite. It's an amber colored kind of oval mite. This is a female adult, and on the back you can see this kind of hour shaped marking. This is the broad mite. This is in the family Tarsinemidae. Just to show you some other mites that are related to it, the honeybee mite is found inside the breathing tube of honeybees. And here's the breathing tube here, and the mite is inside. So you can see if you get enough of those, it's kind of like having phlegm in your throat. It really cuts down on your ability to breathe. Cyclamen mites are closely related. These are more clear white mites, and the eggs are oval shaped and almost look like a small predatory mite egg. They're about half the size of a predatory mite egg, and they're oval or football shaped. The broad mite is amber colored compared to the cyclamen, which is white. Has the hourglass shape on the back, and it has oval eggs similar size to the cyclamen mite, but it has these white spots on it, and some people say there's 29 of them. I haven't really counted them. It has a worldwide distribution, all those black dots on the map. It was originally a tropical and subtropical mite. Preferred greenhouse. It's a greenhouse pest in more temperate or cooler locations in the world. But recently, like in Arkansas, we've been finding it attacking crops like blackberry, and it seems to overwinter here. Some of the other locations where the broad mite is causing damage, it was first seen on blackberry and primocane producing blackberry in 2006 in Fayetteville. And you can see we've been monitoring broad mite in several locations in Arkansas, those red stars. 2017, it was on blackberries in Arkansas, California, Illinois, Indiana, Maryland, North Carolina, South Carolina, Pennsylvania, and Virginia, all those blue spots. And also it's found on other crops in those same states and adjacent states. The life cycle, and I'll show you how to identify the different stages. So it starts out as a spotted egg on the underside of the leaf. Then you get these 6-legged larvae, and they develop into nymphs, which I'll show you in the next slide. And then eventually they become a male, adult male, or adult female. And here you can see a male that has attached a proto-nymph female to its back and carries her around. And we think this may be how they're dispersing from plant to plant, or at least from leaf to leaf. And once the female emerges as an adult, the male mates with her. So that's a resting nymph and a male. So it takes 6 to 14 days to complete that life cycle from egg to mating adults. And each adult female lays 1 to 5 eggs per day, and she may live 1 to 2 weeks. So there's a high potential to produce quite a few eggs per female. Overwintering, Jessica's looking into this. She's been looking at dividing, she collects samples of canes and leaves and leaf litter and soil every 2 weeks for the whole year. And she's been using a Berlese funnel, which is basically just a funnel with the sample on a screen up above and a little light above that, and the mites will eventually fall down to a collecting solution at the base of the funnel. She's looking at leaf litter, pieces of cane, pieces of leaf, and this is what she found in terms of mean numbers of P. latus, which is the broad mite, per sample type. In the sample types, leaves, not very many buds, and canes, not very many. Leaf litter, that's where she finds most of the broad mites, and in soil she doesn't find a whole lot. Some broad mite damage, it does reduce yield, and here you can see cannabis, pepper. It tends to affect the terminals of all these plants. Citrus, it also causes scarring on citrus fruit, impatiens. So it's not only food crops but also ornamental plants. Here you can see a healthy plant in an early summer. The terminal leaves are opening fine, okay, and they're nice color. In comparison, you can see the terminals, they've already lost some leaves in some cases. They look stunted, thickened leaves, they're cupping upward, and they look bronzed. Late summer, you get some, in the left row column here, you get some stunting of the flowers, cupping of the leaves, downward cupping, and also stunting of the leaves. In the second column, you can see what a good flower looks like. These are all the same size. What normal leaves look like, healthy leaves, and here's a trifoliate showing much larger leaf when it's healthy. So terminal flower buds and leaves are stunted, bronzed, and thickened. Under a shade cloth, we've seen primocane plants do this. It's more humid under there, and this is what the primocane laterals look like when they're healthy. This is where some of the fruit is. You'll cut off the tip, and it causes laterals to come out, and then they produce fruit. So that's a healthy lateral. But sometimes when you get high humidity and broad mites present, you get them feeding on these terminals, and they start dying due to some kind of disease, I think. You also, later in the season, get this downward cupping. And sometimes you will see This is outside, not under a shade cloth. We've seen this at Clarksville Fruit Station. We get some of these leaves just dying. And this section right here had broad mites for a while, and now they're mostly on the tip here, but those leaves went ahead, must have had some conditions out there. I don't know if it's rain or what, but every once in a while we get this intermediate branch leaf dying. We're trying to figure out ways to sample for broad mites so we can make decisions whether to spray or not. So initially we sample visible hot spots. We can find that there'll be one cane or one plant in a whole planting that starts showing this bronzing. Then we collect and check about 30 leaflets, 30 of the second expanded leaflet. And here you can see a bunch of leaflets that we've collected, or actually leaves, second expanded leaflet. We note if there's 5 mites per leaf on there. Here you can see the leaflet that we actually take. We don't take these leaves because they're still expanding and will not have as many as mites on them. But we collect this leaf here, and then we just cut off this leaflet and we look at it under the scope like this, underside up. We put this 1/4 inch screen over it, and then we count the number of mites in that, each of 6 of those 1/4 inch screens. And here's the microscope and the counter. Sometimes if the mite numbers are less than 5 mites per leaflet, we can count the whole leaf. But once it gets above 5 mites, we usually use this screen system to count. It's just too many mites. We looked at methods of for efficacy or effectiveness studies of different miticides. This just shows our Petri dish lab bioassay. We dip mite-infested leaflets for 5 seconds in a treatment solution. We place them on the damp sponge. Here you can see a close-up. And then after 24 hours and 72 hours, we look at, is there effect of water on the mites, or agromec, or impede, microthiol, Jameson, stylet oil, trilogy, qualaha, movento, penetrate, or zeal. And then we compare the results. Okay, our broad mites present. We can go to that damaged plant and we check some of those terminal leaves and we look for the female. And usually on a damaged plant you'll start seeing first signs of eggs being laid. This is an electron micrograph of an egg showing those spots. I have no idea what those spots are used for, but they're there. So broad mite numbers on Primark primicanes. So by the 1st of April we pruned the floricanes to the ground, and then any of the primicanes that come up, this is what we were finding in Searcy in 2015 with our weekly samples, or maybe bi-weekly. Here's our sampling dates on the x-axis and the number of predators per leaflet, predatory mites, because they feed on broad mites, maybe. And then in brown, it's the number of broad mites per leaflet. So the brown lines are broad mites or broad mite eggs with the circle, and the blue lines either with the blue square, which are predatory eggs, or just the blue line are predatory mites. You can see most of our predatory mites were out there in April. And then lower numbers were present in June. We would have expected, as the broad mite numbers were increasing in late May, and then in June, we would have expected to see these predator mite numbers go up in response to more food. So they'd be laying more eggs and producing more predatory mites. But I think our native mite predators are just not interested in broad mites, so the numbers didn't go up. But it was right at about in mid to late June that we started seeing bronzed leaves, and at that time we had just increased above, between this sample date and that one, we increased above 5 mites per leaflet. So we're thinking somewhere above 5 mites per leaflet is when you start seeing bronzing. And we're also in primocane, we're near bloom at that point. So native predatory mites, the blue line, don't seem to control broad mite. Just to show you how numbers develop in a field over time, our first hotspot, this is a field, this is a 10-foot, I think it's about a 10 to 15-foot plot noted by each of these little rectangles. This is a row here. We have 5 rows, and then we have actually 12 plots here. This is a plot with a similar unsprayed plot between each of these. So we have 6 plots here. And only that one end plot had broad mites at that time, about 0.2 mites per leaf on the 26th of May. About 2 weeks later, we were averaging, for all these spots here, all these plots, we were averaging 4.8 mites per leaf. We were starting to see some damage in this hot spot of mites in that corner plot. By 6/29, 2 weeks later, we were averaging well above 5.8 mites per leaflet. Here you can see the broad mites are found throughout the planting. By first week of July, we were averaging 50 mites per leaf in almost all the plots we were measuring. So, and we were seeing damage everywhere in anything like above 5 mites per leaf, we were seeing damage in those plots especially by the 7th of July. At that time, we told the grower, I think you need to spray the whole thing. So he sprayed it with Agrimec the next week. So that gets us into how do we prevent damage. So we watch for the first bronze terminal damage, that hot spot in your planting. It seems to usually show up maybe in very few plants in one location. We start sampling for broad mites to see if they're present in those on the terminal leaflets of the damaged plants. Usually they are. Then if the hot spot has more than 5 broad mites per primocane leaflet, we apply miticide usually to the whole planting. Compounds that we have found are effective against broad mites from our efficacy lab bioassay. Here you can see the compounds in the x-axis. You can see just using water alone had a little bit of effect on broad mites, but not much. All these others do have a significant effect on broad mites. And here you can see from 0 to 100% mortality is the y-axis. And the blue bars are 24 hours mortality at 24 hours, and green bars mortality at 72 hours. This gray box denotes any of the compounds that had effectiveness against broad mite that caused more than 80% mortality after 72 hours. So you can see Quillaha, Trilogy, Stylet Oil, Impede, Microthiol, and Agrimec were all effective. So potential of biological control by predatory mites. We were interested in seeing if that would work on broad mites and blackberry. And here's the teardrop-shaped predator mite feeding on either a little thrips or a 2-spotted spider mite. And in some other crops, the predatory mites did delay broad mite buildup if you put them out early enough, when they were between 2 and 5 mites per leaf. So one of the species was Neocelis cucumeris, and it comes in little sachets. For $66 you can get 200 sachets, and about every 10 feet of row we put out one of those sachets. And this is what it looks like. It attaches to a cane. We usually try to put it, we released them near the terminal because that's where all the broad mites are. So we tried to, and then what happens inside this sachet are predatory mites and also a soil mite that they feed on. that they reproduce on. And then they put a little hole in this sachet, and the predatory mites will come out and move on to the plant. So what we're doing here, we're seeing that at Circe, we're getting some delay of our first mite peak. That's noted by this red arrow pointing down. In each of our slides here, these are sample dates on the x-axis, and on the left y-axis is the number of broad mites per leaflet, and the right axis, y-axis, the number of predatory mites per leaflet. And the predators are noticed as the black line with the triangles, and all the different stages or total active broad mites noted by PL are noted by just the colored lines. So you can see if we did nothing in a plot, you can see we had predatory mites there all season long. And we had buildup of broad mites in June, and then definitely in July. Then in this plot, the grower sprayed on about I think after July 7th. So that shows you what happens if you do nothing until July 7th anyway. And he sprayed July 7th for all these plots, all these graphs. The next one we released SWD, which was expected to, which is known to feed on broad mites. And you can see after the release on 5/20, we did have some predatory mites, but the numbers went down because we didn't have any. It looked like they were causing some reduction in broad mites, because our broad mite numbers were low in May and June, early June. Then they started increasing after mid-June, and our predatory mite numbers also increased, kind of following the broad mite numbers. Then they again peaked in. Here you can see our different peaks. Then we used sulfur alone a couple times, May 7th and 20th. And you can see the May 7th and 20th sprays really reduced all mite numbers, predator mites as well as broad mites. So we didn't have any broad mites till almost July 1 after our May 20th application. And then of course we sprayed after July 7th. Then Cucumerus was applied on May 20th, and you can see we had Cucumerus out there. It feeds on other things, probably like thrips. And it delayed broad mites until almost July 1, just like sulfur. So it looked like it was doing a little bit better job than SWD that didn't really knock down numbers as well in May and June as Cucumerus did. We also did some similar treatments in Hope, Arkansas. We had the control, which is, these are our dates of sampling on the x-axis. The y-axis is just broad mites per leaflet. And then the dotted black line is a control situation, basically how broad mites build up over time in the high tunnel. And then the green line shows what happens when you use sulfur plus Cucumerus, which was supposed to be fairly good. And then sulfur alone, 2 applications of that on the 20th and 7/1, you can see it delayed emergence of broad mites pretty well compared to the black line. But it seemed like everything by July 1, the broad mites do come out, get pretty high, as high as the unsprayed control. But basically we're getting a 2 to 3 week delay in mite buildup using sulfur. Now to show you what the effect of Agromec is, here's our 7-2 or week 8 plot of mite numbers back in Circe again. We applied Agromec on 7-15. This is what happened by 7-21. You can see it went from hundreds of mites per leaf in the blackberry plants to just that one little hot spot again. Everything else is pretty well controlled. It probably just didn't get a good turn on the row and turn the sprayer on well enough there, but it even that spot had lower mites. So Agromec were compounds that we're now suggesting. Agromec is our only conventional compound. The others are all OMRI approved, but they all have, these are all with different modes of action, as you can see in the right column. So recommended miticide program in May, put out a spray, maybe 2 separate sprays, 1 week apart of microthiol or MP'd or JMS styloid oil. And this is as long as the temperatures stay below 90 degrees following the spray or during the spray. July and August, if you start seeing some damaged plants, you can apply Agromec when it's warmer. Usually then it's above this 90 degree threshold for the other compounds. So you can apply Agromec, and maybe a second spray of Agromec is applied a month later or 2 months later if you have mites reappear. So that's all we know about spotted wing and broad mites. And we did have some funding from the USDA Hatch project and also a SARE project, Southern SARE, Sustainable Ag Research and Extension. Then we had some funding from Southern Region Small Fruit Consortium in IR4 for the efficacy work. And we had a specialty crop block grant from Arkansas Ag Department and Extension IPM. We had technical assistance and also appreciate the growers. If there's any questions, I'll try to address them both on spotted wing or broad mite at this time.